Hi, my name is Roland Wilhelm. I'm a student at UBC, University of British Columbia, and I'm studying the forest biome in the northern climates, the temperate forest. And we study the microbial community that lives in the forest soil that breaks down cellulose and lignin, so the plant cell wall, the woody debris. And it's important because these forests and these ecosystems collect and trap a lot of the CO2 from the upper atmosphere, as well as provide a lot of the nutrient cycling for all of the plant and animal life in the forests. So what we've done is used a very novel approach where we label cellulose with 13C carbon. So the carbon-13 is a stable isotope of carbon, and because the compounds we provide the soil are enriched, about 90% carbon-13, we can track that through the microbes that start to break that down and incorporate it into their biomass. So by doing that, we're able to track which organisms are incorporating into their DNA, and we're then able to sequence that DNA. So there's one way of identifying the organisms that are in charge or responsible for breaking down cellulose and decomposing the plant matter in forest soils. But we're also able to track how fast that happens with that same technique. So you're tracing or tracking the carbon-13 through the ecosystem, and that gives you an idea of how at different rates, where it's ending up, and the fate of the carbon. So we're able to track the carbon as well as who's degrading it. And so what we found was, in comparing different forests that had been harvested with different forestry techniques. So we had an unharvested control, and then we had different layers of timber removal. So we had when the whole tree was removed, when the whole tree plus the branches removed, plus the whole tree and topsoil removed. So these are different practices that are common, more or less common in parts of Canada. And we found that this did influence the rate of decomposition of cellulose, so that was one of our findings. And then also the diversity of organisms. So that's not necessarily anything breaking that anthropological or anthropogenic disturbances reduce or affect diversity of organisms on the planet. We've known that in macroecology for a while, but this is interesting because it's affecting the microbial communities, which is sort of the foundations of a lot of the nutrient cycling. You see that the decay of plant matter builds up and builds up, so it's sort of the source of a lot of the energy and biomass in this ecosystem. And then also, the other sort of novel aspect of the research was that we were able to identify the organisms that are involved, and we found quite a number of novel groups of organisms that had never previously been known to degrade cellulose. So there's the possibility of then looking deeper at these organisms and discovering new ways in which cellulose is broken down, new enzymes or new approaches that could be used in conversion of biomass into biofuels or other raw chemistry that we can use. So I think that sums it up.